Kia ora, everyone. Thank you all for being here today. We have a few faces here in Tauranga and also online, so thank you very much for joining today's ERI seminar. And I would like to introduce to you a former colleague of mine, Mirko Wölfeschneider. He studied biology at the university, the Philipps University in Marburg, which is central landlocked Germany, where he studied, I guess, rivers and freshwater systems, or the biology therein. But then he went to Giglio, a tiny little island in Italy, and he started diving. So he converted to the other side, to the marine side. And where I met him was during our master's at the University of Bremen in Germany, in the north, where we did the international studies in tropical marine ecology together. And that's where Mirko fell in love with the mangroves. And today he will also tell us a little bit more about blue carbon and mangrove systems. He had extensive studies in different parts of our beautiful planet. So I'm quite excited to hear about probably Brazil, I think today will be on screen. And without further ado, I'm just gonna share our screen with Mirko's presentation. Please let us know online if there's any trouble. I will make sure to keep an eye on the chat as well. And once Mirko is done, I'm happy to take questions. So hopefully we should be done in about 45 minutes. And if people have time to stay for a few questions, we're more than happy to take them as well. All right. I hope that you can all see this. And Mirko, over to you. Thank you very much.
Thank you very much, Natalie, for this very kind introduction. I'm very happy to be here. It's really exciting. And yes, so you can see already on the screen the title of the talk. So it's about blue carbon coastal vegetated systems, and especially the organic metadynamics and how to trace them, how to better understand them, and all of this in a frame of one of our current projects, the Sea4Society project. And this is a super long title, and I would like to start actually at the end here with introducing very briefly our cool project. So Sea4Society, this is about searching solutions for carbon sequestration in coastal ecosystems. And this is all about trying to make a change with current problems we are facing in terms of high levels of carbon dioxide in the atmosphere. Yes, and this is what our main project is about. And I really like this because this kind of binds together my interest of making a change in the world, leaving a good impact, and working with the beloved ecosystems you can see here in the background already. So very briefly, why is this important? I guess you all know this, you're all scientists. Just want to quickly show this again. So we have a few different, I hope you can all see the cursor here, a few different trajectories. The red one here is if we would just keep on going, then predictions say we end up around 4 degrees more plus in global average temperatures. If current policies, the orange one down here, would take place properly. So that would mean we are around ±3 degrees Celsius. That here is the climate convention from Paris, around about 2.5%. And then down here, we would near kind of the optimal 0 or even below 0 emissions. And for this, we need to reduce carbon emissions, definitely. But to reach a stage here where we can sequester carbon again from the atmosphere, carbon dioxide, remove it from the atmosphere, we would definitely need something to assist us with this. And yeah, this is where our big research mission comes into play. This is actually only half of it in the marine realm. We also have a big finance support by the German government, which also looks at the terrestrial part. But these are the 6 projects which look into the marine area. And they look at very different various things. You can see a very technical solutions, introducing CO2 in different rock types or changing alkalinity in the water to sequester carbon dioxide. And then we have here two more nature-based solutions. This is a mix of nature and technology. And here is our project Sea for Society, which, well, focuses on carbon ecosystem in the coastal areas. And what are we working with? We're working with kelp forests. You can see in the top left corner, or kelp beds in this case. Then we have the beloved mangrove forests, which my strongest focus is. But we also work with seagrass meadows and salt marshes, which are all in the coastal region and sequester carbon in their biomass and also in the sediments associated with these systems. And why are we doing this? Well, because if you look at this, on the right side here are 3 of the 4 systems we're looking in. And older studies have already suggested there is a very great potential compared to terrestrial ecosystems in terms of carbon storage. In this case, annually per year. But you can also see that here with the big variation here indicated by the bars, there is still some research that needs to be done to identify this a bit better. Here are a few numbers to this, which is one of the outcomes of the literature research in our project. And you can see that among the most productive or effective ones are salt marshes and mangrove forests. But you can also very clearly see here, which I want to point out a little bit, the big range of data available at the moment, because there are actually only very few measurements worldwide where extrapolations are based on. So there is definitely room to improve. But how is this happening now? How is organic matter produced and then also carbon sequestered in the systems? So it all starts with a plant. In this case, I'm going to show you exemplary from the mangrove system. And these plants, of course, do photosynthesis or also the algae, and then they use up CO2 from the atmosphere or the water, and then this is incorporated in the biomass. Over time, this biomass, in this case, is an exemplary the leaves. They will drop from our mangroves here and will fall down on the ground and be eventually incorporated as autochthonous particulate organic matter by sediment slowly being deposited above them and then incorporated and stay there on a relatively long time scale. But also there is then in the marine realm, the tidal influence, which not only brings in allochthonous particular organic carbon, which can be then, for instance, seagrass leaves, algae, tally, or whatsoever, but it can also flush out part of the autochthonous in this system produced organic matter into other systems which are nearby or maybe even very far away. What happens inside the sediment is there are degradation processes taking place. We have microbial, but also physiochemical processes which alter the organic matter. Some of the part will be transformed into dissolved organic matter, so chemical compounds, which are being washed out. It's kind of similar if you think about the little tea bag you put in your tea in the morning, maybe. And then, of course, the part is also released in the greenhouse gas emission. So all these different parts we investigate in Sea for Society, all the different steps. But there is a trick to it. So, for instance, how do we manage to differentiate between allochthonous and autochthonous organic carbon? This is often important in terms of stability. If organic matter is traveled from far away and deposited, it's already pre-degraded versus the relatively fresh autochthonous organic matter, which is then incorporated in the sediment and behaves a bit different. Traditionally, or in the past, often people use stable isotope analysis for this, which works in very simple systems, but it can become quickly very, very tricky when you have very complex systems, where you have very various different primary producers, so the models then and the calculations getting a bit iffy. Another problem is how the CO2 is incorporated. Mangroves have a photosynthesis pathway, which is very similar to terrestrial plants. And also some of the seagrass species can be hardly differentiated in some systems between each other. And then you have this pool and can say, okay, this is maybe mangrove, maybe terrestrial, or maybe any other organic matter. And this is actually where I'm working on in the project and where I have been working in the past on. And this is what I want to understand a bit better. So how they exchange this, how the dynamics is, to maybe in the future get a bit more elaborated flux calculations for this very complex coastal ecosystems in which we're working. And therefore I brought you a case study. And this case study took place in Brazil. On the eastern part of the country, here on the coast, there is the state of Bahia and the city of Salvador, and right next to it is an estuary of the Jaguaripe River, you can see here. And what you can see here on the map, these are 5 transects where I sampled along a salinity gradient to try and see if I can figure out which organic carbon that occurs in the system is from which source. So what you can see here on the map, we have this greenish parts. This is all mangrove forest, which is documented. And also here, we have in this part of the estuary area, which is connected to a bigger bay system up here, we can find seagrasses, which grow there relatively abundantly. So what did I actually do? I went there and I sampled along the 5 transects, along the salinity gradient, and I did this twice. Once in the rainy season, once in the dry season. And in this study, which I'm presenting today, I focused on the upper 1 centimeter of the sediments. And then to get an idea about the organic matter and have the ability to identify molecular markers to be able to trace them in the sediment, I also took reference samples from 30 abundant plant species around the area, certainly the mangroves, the seagrasses, but also macroalgae and quite a number of terrestrial plants which grow along the river, and then leaves and other material also end up in the system of the estuary. The samples then were simply freeze-dried and milled. And then, in this case, for the particulate organic matter, analyzed by pyrolysis gas chromatography and mass spectrometry, which is an analysis which, for those of you who might be not so familiar with it, enables us for once to not specifically extract a certain group of chemicals for like liquid injection chromatography, but it burns all the material in there, and then the gas is released from that. The pyrolates will be then analyzed in the gas chromatography, and later on it's coupled with mass spectrometry to identify the weights of the ion masses of the different compounds to get an idea which compounds might be hiding behind this. So that's for the theory. Now, what did we find? And this is a PCOA plot here with Bray-Curtis and the similarity matrix in the background. And it shows us from our different sources which ones are close to each other and which ones are a bit more away from each other in terms of chemical composition. So what you can see here, this big triangular shape, these are our terrestrial plants. Here are macroalgae, here are macrophytes, which we sampled upriver, seagrasses down here, and here are mangroves. And this picture also describes very nicely what often happens with stable isotope analysis. You have quite an overlap here between terrestrial plants and the mangroves. So when I saw this plot the first time, I was like, well, that didn't work as planned. So I cannot really differentiate between mangroves and terrestrial plants. And then I was like, really, oh man, why did I spend all this time of my life working on this? But then on the bright side, okay, I thought like, let's look a bit more in detail into this. And then I ran a few analytical steps for this. And then I could see that terrestrial plants and seagrasses, so terrestrial plants here, seagrasses there, can be differentiated. That's already cool. It's, however, not so easily possible for macrophytes and terrestrial plants, which, no surprise, they're relatively close to each other. Then I thought, okay, let's have a look for the mangroves and the terrestrial plants. And it actually seems there is some evidence that it might be possible if we look a bit more into detail. So I thought, okay, that's at least a glimpse of hope, right? And then the mangroves and the macroalgae, again, they are very easily distinguishable here. And also the seagrasses were quite different. So I thought, okay, maybe there's hope to somehow find chemical markers to identify our different groups, functional groups in this case, of primary producers. And then I looked into the whole data set and identified which chemical compounds or markers are specific for which of the groups. And this graphic looks very big, but don't worry about the numbers. So the important numbers are all the 5 numbers are out here, because these are chemical markers which in this setting, which what I sampled, and in this estuary are markers which are highly unique for only this functional group. Mangroves, terrestrial plants, seagrasses, macroalgae, and macrophytes. And then I got already a bit more excited because it showed me that the mangroves in total, the different species there, share 36 chemical markers which are unique for them. That means if we find them in the sediment, then we can be pretty sure that mangroves are around. So, and then I looked a bit more into detail into this and split them up. We have 3 mangrove species in the area. 1 laguncularia species, 1 avicennia species, and 1 rhizophora species. And you can see here that, well, Rhizophora manglae has by far the largest number of identifiers, which is something, but also for laguncularia and avicennia shaweriana, we have a few markers which we can identify only for the species. So if we find either of these specific markers, which are highlighted in green up here in the bar plot, then we can be relatively sure that, okay, here in this organic matter pool, whatever we analyze, filters, sediments in this case, which comes a bit later, we have these species contributing to the organic matter stock in this particular field where we're looking at. And this is something really cool because this allows us not only to go into groupings of different individual species, like for instance, the mangroves, but we can even go down to the species level. And that was then something I was really happy and excited about it. And then I thought, okay, so we find them in the leaves, do we find them in the sediment? Because if not, then all of this is a waste of time. So I looked into it and we actually found them. Or I actually found them and could identify them. What you can see here on the scaling, this is a normalized scale. So from what I found to what I not found, I just normalized it between 1 and 0 to make it a bit more visually understandable, or more easily understandable in the visual depiction here. And yeah, this is for the dry season, the results. And here I just plotted for the sediment samples we took, the values from the normalized identification process of the 36 markers and had a look which ones we can find where. As a bit of a side note, here you can see that these mangrove markers, I put them all together. What is hiding behind it is this, the majority of them are actually from Rhizophora manglae, which is for one not surprising because they have, if you look back here, they have the most uniquely identifiable markers, but also Rhizophora manglae is one of the most dominant species in this area. Laguncularia is normally a fringing species or a pioneer species in the early stages of aggregation of sediments in the area, and Avicennia shaweriana comes in between, and the climax stage of the forest is Rhizophora manglae. So that's how it is. But it also shows really nicely that here, where there is the main mangrove fringing area, like up until here, transect 3, we have a little bit here and a few single trees here on transect 4. You can see we have a very high value here for the identified markers in this area, and then it slowly fades away. And already on the transects here, transect 5, we could not even find the markers anymore, which is interesting and which kind of overlays with quite a few studies of colleagues from our institute, which said, if you go further off shelf, it's really hard to still trace back mangrove organic matter. And the reason is probably because the dynamics strongly keep the organic matter in the area, depending on the tides, the water flow, and it kind of sticks around close by and doesn't necessarily travel as far, depending on the setup here of your system. And then when it comes to the rainy season, you can see a bit of a difference here. And you can see here, due to the high precipitation, which happens not only in the hinterlands, but of course also here, and flushes then with surface runoffs more organic matter into the system and has a more outflow of the riverine parts here, you can see that the organic matter travels further out. So you can see here much clearer signals now. Where's the mouse? Going down to transect 4 here, even out, and getting stronger down here. Again, transect 5, we couldn't really find much anymore. And here, transect 5, the northern point, yeah, this unfortunately, we couldn't recover any sediment due to strong waves, but we assume that it's very similar to the rest here, that there is no more organic matter found anymore. So this was really cool. And this can be done for all the other 4 different groups I identified with different values, but I want to keep it for the mangroves here because mangroves is what I love to work with. So I thought, okay, we keep it with here. So I was then really excited, and this could be potentially, we still have to refine a bit to get a very nice index, which can be globally applied. But this is the first study, to my knowledge, which worked on this. So I'm pretty cool that it worked so far. It still needs some refinement in the future, but I think the potential is pretty cool. So this is the one part. If you remember here, we have now looked at the particulate organic matter. But then the other part is the dissolved organic matter. And this is also what was studied by me there. And here you can see it was also sampled along the 5 transects here, along the salinity gradient, but then I took further samples here in the outer S3 part, and then here in the river, very upstream river part, where we have in the rainy season, definitely pure freshwater input. In the dry season, sometimes you can get a hint of seawater encroaching up here, but normally you would also maximum reach salinity of 1. These are taken additionally for the reference samples you will see later on, which I come to in a bit. And then the sampling strategy, well, it's pretty much similar along the transects, along the gradient in rainy and dry season. It was surface water taken for this study, which I'm presenting here, and also additionally environmental parameters such as salinity, dissolved oxygen, pH. Yes. Then it's not so easy to get markers from, let's say, mangroves. And what I looked in specifically in my study is what leaches out from mangrove leaves. Because mangrove leaves, if you have been to mangroves already, they always float on the surface. They go back and forth, being pulled out and pushed in by the tides. And there is, like in your tea in the morning, material leaching out, right? So this is what I looked at, and I created something which we often refer to as mangrove leaf tea. So we collected a bunch of mangrove leaves, senescent mangrove leaves of the different species, put them into distilled water to leach out, and then measured these as our reference samples to work with later on. Then we analyzed carbon and nitrogen, which were filtered and then acidified, in this case, organic carbon and nitrogen. And then we also ran it through the FTIC-RMS, which is Fourier transform ion cyclotron resonance mass spectrometry. Which is a big word. It's a process which gives you a similar output to a gas chromatograph coupled with mass spectrometry, but the measuring part of the cyclotron resonance detector here works a bit different by charged ions being resonated in a system, and then by this resonance-induced electrical current being measured, then what actually is there and how strong the ionization works there, coupled with the mass spectrometry. In the end, you get a really high resolution output, being able to identify some formulas for the compounds which you're looking at. Yeah, same question then, what did we actually find? So here the plot looks different, and it's a bit differently depicted here. What you can see here are not the reference samples, these are the actual water samples from the estuary. And I have cut this into two different groups. Here on the left side, the legend is on the second part, you see the mangrove fringe parts, transects 1 till 3, if you remember, the more upstream part. And on the right side, the purplish things, these are here and also here. These are the more downstream areas of the estuary where there is almost no mangrove fringing happening anymore. And you can see here they are very well differentiated. So you can see mangrove fringe, not mangrove fringe, dry, and here rainy season, mangrove fringe, not mangrove fringe. They are very clearly distinct. A bit better in the rainy season than in the dry season, but nevertheless, you can see the differentiation. And then what also happened here in this plot, you have different parameters which I took, incorporated in this graphical displacement here to see, okay, what can be seen or what are differences associated with this collection of different spectra and formula accumulated within the samples. And one thing which is very unique for the mangrove fringe transects is that we have a high dissolved organic carbon level, a high dissolved nitrogen level, and we have a lot of compounds with sulfuric components with double bonds. So very stable, actually, components of the dissolved organic matter, and we have aromatic components. And especially the sulfur-containing components are a very good hint for processes which happen in anoxic sediments, especially mangrove sediments. And then in the other transects, which are not so strongly mangrove fringed or not at all, we have a strong correlation with salinity, dissolved oxygen, pH, compounds with nitrogen, which is more coming in from the ocean and associated with phytoplankton from primary studies, and then phosphate and high hydrogen carbon ratios. Yes, so this already was pretty cool that you could see such a clear differentiation between them. But then I thought, okay, can we then identify specific traces for the mangroves which I leached with the mangrove leaves? And if I show a similar graph displaying the sources, so I differentiated here between the mangrove leaf leach, the riverine, and the oceanic. The oceanic was the outer estuary sample, the riverine sample, and the mangrove leaf leach. And you could see that there is a certain pool for each, which are the outer numbers here again, for each group, which are unique markers which could be used later on. This applies here for the dry season, 421 markers for the mangroves, and also for the rainy season, which are even a bit more, 489 chemical markers, which you could identify for the mangrove leaf to trace them later on. And then one thing I have to introduce you to are the indices we evaluated to make it a bit more standardizable and reproducible across the world. If you do a study in a different estuary, for instance, you have your target components here in the upper part, which are 40 peaks we identified with different characteristics, very specific for the mangrove leafage. And then this will be divided by the sum of the to be traced target groups and the reference peaks, which come from the Island of Stability, which is a study which was conducted in 2004, if I'm not mistaken, and looked into various marine samples and identified a group of 281 molecular formulas which were present in all marine samples. So with this, you can get a relativized index for our mangrove leafage and can apply this also for other samples, which you take on another end of the world. And then we thought, okay, our colleagues also work on a similar index for mangrove pore water derived markers. So we thought, okay, why not apply this and make it a test and see what are the results? Are there similarities? Are there differences? And this is what can be seen here. This for now is the mangrove pore water. And you can see here, the mangrove pore water seems to be strongest, of course, on the upper three transects. No surprise, this also the PCOA plots indicated that there is a difference. And then down here as well, it's getting lower, but it's even washed out here into the outer estuary and then the coastal waters and traveling on. One thing which was a bit, we worried about it in the beginning, but then we thought, why could this be? We have the highest value up here in the green side. And this we assume is due to that we have extensive macrophyte areas here. So this is a grass species which can also persist into submerged areas and grow there very strongly. And this is very common here in the upper, relatively shallow part of the river. So there is also anoxic sediments, which, similar to mangroves, have probably similar signatures in terms of high levels of sulfur content and other markers. Nevertheless, it seems to be working quite well. You just have to be cautious if you have other systems with similar characteristics in the area. And then we look at the mangrove leafage. And here you can see a pattern which is actually relatively similar. You have a strong points here in the upper three transects and then less strong indice values in the lower parts. And one thing which unfortunately was not in the protocol to record, I just have pictures for this. We had observations here that in this specific areas here, up on the left, on the northern side, here in the middle side, and here in the middle and north, there were always accumulations of mangrove leaves floating on the surface. And they went even out here to the outer estuary and built this huge meadows of floating mangrove leaves on the water surface. We have a picture actually where we take samples right next to them. And this, I would assume, explains this very high value outside in the estuary, which was quite interesting for us because these compounds are normally relatively labile, but you can see they are being transported if the conditions are matching to even in the coastal waters, and then can there be used up by microbes and fewer phytoplankton or whatsoever. This was in the dry season, and in the rainy season, it's relatively similar. You can see here again, like also for the sediments in the rainy season, the organic matter, in this case, it dissolved, seems to be traveling further outside of the estuary, seems to be pushed more out because just the river runoff is higher. And this is also reflected here in the index for our mangrove leafages. And you can see here that the values seem to be a bit higher overall. And this we assume is due to the fact that, of course, the runoff of precipitation here in the mangrove forest also flushes out mangrove leaves, which then leach into the surface waters. So yes, what can we learn from this? Or why do I find this so exciting and I'm really happy about the study? To enhance CO2 capturing capacity, we need to understand blue carbon ecosystems and especially the dynamics there. At the moment, we mainly consider only allochthonous organic carbon being stored in the sediment and ignore the autochthonous part, because we just simply don't understand it to an extent that we can really make precise calculations about it. But to fully understand this, we definitely need better tracing capabilities of our analysis, and the two methods I showed you, or the two approaches I showed you, might be some way to go in the future and can be also applied to various other systems and other details. And yes, with this method, as I showed you, it's even possible to go beyond functional groups. You can even, in some cases, go to species levels, which I found really cool because there are not many besides DNA or eDNA techniques who can do that. What in the end needs to be done, this method is more on the qualitative part of the spectra, and to make comprehensive calculations for carbon fluxes, we are currently on the way to refining this to a more quantitative approach by combining it with various other methods. And yeah, that's what I'm doing at the moment. I probably will be doing in the future. And with that, I want to say thank you for your attention. It's a little bit exciting, and yeah.
Thank you very much, Mirko. We would like to ask anyone if if you have any questions, either on Zoom or also here in the room. We're happy to take them now, just before wrapping up. Are there any questions for Mirko?
Yeah, thanks, Mirko. I was wondering about the sampling, your sampling strategy in the POM part that you talked about, how it looked like you had many more samples for the terrestrial plants than for all the others. And then actually also, if you go a little bit further into, and that's reflected, sorry, in the Venn diagram, that's reflected in kind of like the specific markers for that group, right? And actually, if you looked at the different individual components, it seemed like the sampling depth was very well correlated with the number of markers that you got in each of the groups. And then going further in, if you look at this mangrove species, you said the rhizophora species were the most prominent in the area. They were also the ones that showed the most markers. I was wondering, how do you have any idea of how deep you would have to sample to kind of like reach a saturation point of where you can say you've maybe sampled all of the possible markers? That could be a good kind of like indicator for sampling depth in the future.
Yes. So maybe let me go to the first question. So there is definitely this queuing of the results by sampling, of course, more terrestrial plants. The thing is, for once, we have the problem that there are no more mangroves in the area. So this is something we are currently working on. We have a huge variety of samples taken from other parts of the world, which ideally we can incorporate into this. But also this is something where we say this is, besides a bit of the part of the analytical method itself, it's more the qualitative, because we haven't found a good way to incorporate this into our analysis to get this unbalanced results kind of leveled and being able to then fully quantify the outputs. That's why it's at the moment more on the qualitative side, but this is definitely an issue we also saw already and we're working on in the backgrounds in our labs at the moment, but we have not found a fully pleasing way to change that yet. Because of course, if you sample more, you have a greater variety, so you might end up with more parts, more compounds, which are represented in a group where you have a larger sample size. This is something we're working on at the moment. For the second question, the assumption I have, or what I discussed with my colleagues and peers on that, is that the Rhizophora group is a bit more distinguished from many terrestrial plants in the areas compared to the families and higher hierarchical orders of the Laguncularia and the Avicennia group. So we assume that there is more overlap between these two groups and the terrestrial plants, which then rule out these possible chemical components as markers for the specific species. This is also an issue we're working at the moment, and we are breaking our heads, like how you would say it in Germany, to figure out how we could come up with an index system, for instance, which relevates this or levels this a bit more, to not skew this one or the other direction. Because it's not that we have more markers because Rhizophora mangrove is more abundant there. This is just how the system is, but we measured the same amount of leaves. So this shouldn't be an effect there. But yeah, so this is in a very early stage or relatively early stage of method development, and there are still a variety of things you can do. If anyone is interested, I think our group and my boss is very happy to have brilliant minds around to discuss this. Yes.
Cool. Charles?
Yeah, hi. Thanks, Mirko, for a great presentation. So I have two magic wand questions, right? My first question is, if you had a magic wand that gives you all the knowledge you need, what would that knowledge look like? And then the second magic wand is, if after that first magic wand has worked, what would you wave that wand with and create change? What specific actions would that magic wand allow?
Thank you very much, Charles, for this very nicely phrased questions. So with my first magic wand, I would definitely go and sample every single primary producer in the world and analyze them. Ideally within a day and not within a couple of years. And then to get a very holistic picture of what can be markers really effectively used for what group. And then maybe this would already rule out the unbalance of the system, but it would also maybe increase it even more, and then we would have to see how to deal with it. The second wand, if we would have this baseline knowledge, I would probably wave it and make it a kind of compass to point at the most effective methods to combine it with to quantify it fully. Because there are a few detection limitations in the analytical background, which I didn't go into more detail here with, which also limits us a bit on the quantitativeness of our results there. So one option could be to combine it with, I don't know, maybe stable isotope measurements, and then get really quantitative, really comprehensive data to be able to make global calculations and really understand the system. I don't know, maybe in the lifetime of us, we will achieve this by different groups working together. Maybe it still is a magic wand. Maybe we invent this someday and then we can figure this out. Yeah, and maybe just a more practical thing is then, the idea is what we also have at C4 Society, to then be able to create combined coastal management plans with different systems, not only looking at one system, but for instance, mangroves produce organic matter, it's being flushed out, and then a seagrass meadow captures it, stores it in their sediment, and then we have increased carbon storage. So these are kind of future dreams which go beyond the two ones I have, which maybe would be a third one. But yeah, I hope that answers your questions, Charles. Or, well, three short question, I would call it. And then I see a hand from Hamilton here.
Yeah, so a couple of people sitting around a table, maybe. Is that you guys?
G'day, yeah. I don't know if I've got the wrong end of the stick here, sorry, but with your indices showing the highest impact or the highest influence from mangroves in that river run bit, presumably that's far enough up the river that you know it's not the mangroves that are doing it. So how do you know that the rest of it tailing down as it gets closer to the entrance isn't just a reduction in the influence of the riverine instead of the influence of the mangrove, if that makes sense?
Yes, that definitely makes sense. Yes, this is also being studied there. I mentioned, I unfortunately don't have the graphs at hand right now. I mentioned that we also looked at total dissolved organic carbon in the area. And one thing which further studies from one of my peers showed very early in the beginning of the 2000s, showed that mangroves in areas where we don't have this macrophyte grassy swamp areas upriver excrete a high amount of total dissolved organic carbon into the system. And with mixing plots, which we calculated, or which I calculated, which hopefully will be published in a paper by the end of the year, you can see that if you make a line between the, maybe I go back in slides. Can I go back in slides? So if you calculate a simple mixing model line between this riverine spot and down here, the spot in what we call the outer estuary, which is maybe not the very perfectly right term here. But then you can see that especially in this area, you have a high input, which is not explained by simple linear mixing down here from the freshwater values down to the seawater values. But there is a very strong input here in this specific area where the mangroves reside. So this is a indication, a very strong indication for us that even though you have a similar input by the system up here, because we assume it's also anoxic conditions similar to mangroves, we have a very clear indication that there is even more put on top of the mangroves there. And yeah, this is the thing which we're working as, and we hope we can refine with the traces here. If you have simple systems, only mangroves present there, that would not be an issue at all. And this is a place where this index was developed. But we applied it on a more complex system here, and then you can see it's not perfect. So we're also working on it at the moment. How this target groups are assigned is high level of sulfur atoms in the chemical structure and correlation with barium, if I'm not mistaken, by our colleagues in the lab we work together with. So she correlated these two factors and used the peaks as target peaks for the pore water, which have the highest correlation with these two variables. But as I said, this was more of a trial run here, and we have indication that it's still working, but in more complex system like this here, you might have to carefully rethink what you're doing. And yes, I hope that answers your question.
All right, we might just take one more question just to wrap it up before one. Shami, would you like to read it out? Or?
Yes. Hi, thanks so much, Mirka, for your presentation. It was very interesting. Very nice to see your case study and results. I have a question that kind of pulls out a bit towards the broader kind of blue carbon and carbon marketing kind of space at the moment. And I have a question regarding the need to understand this complexity of carbon dynamics in mangrove ecosystems and today's current practice of carbon accounting for the carbon market. So my question is that blue carbon projects already do exist across the world, in which there are carbon codes and methodologies for carbon accounting that exist. Are you working with any organizations or other universities or firms to kind of implement your research and results into the practice of carbon accountings, or are you aware of any initiatives of this?
Yes. So the clear answer is yes. The project we're working with, C4Society, what we are studying there is, this what I presented there is just a fraction of what we're doing in the project. We're doing much, much more things in there. We are a group of 44 scientists in 8 institutes across Germany, Malaysia, and Colombia. And this is the first phase to get kind of a baseline. From next year on in August, it will be the second phase, which is starting with which is the implementation phase. I actually just came from a workshop in Malaysia before I came here, where we had a stakeholder workshop where we started to initiate a collaboration with them to actually implement our findings and on the same time validate these findings in regrowing systems like replanted mangroves, replanted seagrasses, and where we can find it replanted salt marsh areas or renaturated salt marsh areas. So this is in progress, and we have so far, I think, 5 NGOs on board at the moment. Yes, from Colombia, because the workshop was already in May. As I said, in Malaysia, it was just a week ago. So I came here 3 days ago. And then we also have a partner in Germany on board, which is called Fairgrove, which kind of links local initiatives, for instance, for replanting mangroves with the carbon market, and then also investors finance this. So this is in place. It's happening already in the background. I didn't just want to talk only about this project here, but also of science. So this is happening, and what is a problem, or what we see as a problem at the moment, this carbon accounting often takes only allochthonous material, which I mentioned before, and not autochthonous. And we try to change that a bit, because we have ecosystem coastal areas, which include seagrasses, salt marshes, and mangroves, for instance, together in one area. The effect of carbon, or the capacity of carbon sequestration, seems to increase. And this is not captured by the current systems at the moment. And this is something we try to work on with the work we're doing at the moment. So hopefully soon we will have the first case studies to implement this and then verify what we do.
Thanks so much for your answer.
Yes.
Awesome. Thank you so much, Mirko, for your presentation. And as a token of gratitude from the Environmental Science Research Institute, we give you this little koha, which is like a little trophy to take home.
Thank you.
So thank you everyone for joining on Zoom and also in the room here. And yeah, Mirko's details are just on this last slide, so feel free to get in contact if you have any more questions. Thank you very much.
Thank you.